Good afternoon. Today we are going to deal with the C4 pathway. I will try to explain where the word C4 comes from. Upfront, let me say, all the plants, if they are to make glucose, eventually, they must go through a Calvin cycle. A Calvin cycle is the only cycle that results in carbon dioxide being fixed to make glucose molecules. And the plants that immediately after the carbon dioxide has entered the leaf of the plants goes directly to the Calvin cycle are called C3 plants. So we can say a Calvin cycle is an equivalent of a C3 photosynthetic system. Now we have to deal with the C4 pathway. Why is it called a C4? I will outline here. I will first describe the C4 pathway. This is the leaf of this plant. This pathway takes place within two cells. The first part of this pathway takes place here within the mesophyll cells. And the second part, which is the actual Calvin cycle, you can see there is the CO2 entering the Calvin cycle. The Calvin cycle takes place within the vascular bundle sheath cells. Here, there. This is the vascular bundle sheath cells. So you can see in this plant, when carbon dioxide enters the leaf, it does not immediately enter the Calvin cycle. No. It goes into the Calvin cycle through a very long route. And why is that we'll talk about. But let's see step by step what happens in this C4 plant. There is the carbon dioxide from the atmosphere entering the leaf of the plant through the stomata. There is the stomata. You can see the CO2, this yellow arrow showing you the CO2 coming in. After the CO2 is inside the leaf, this CO2 is going to interact with water and is going to be converted into a bicarbonate, HCO3. So the CO2 is hidden in this bicarbonate, but you can see that bicarbonate has got a charge as opposed to that CO2 which does not have a charge. So there is the CO2 after entering the leaf being converted into the bicarbonate. All this taking place within the mesophyll cells. The next step is going to be this bicarbonate that has got carbon combining with phosphoenol pyruvate, abbreviated as PEP. This PEP has got 3 carbons. There is 1 carbon here in this bicarbonate, as you can see. The product of this reaction, the bicarbonate combining with PEP, it's a compound called OAA, oxaloacetic acid. This is an organic acid. It's an organic acid. The enzyme that catalyzes the reaction in which bicarbonate combines with PEP is called PEP carboxylase enzyme. PEP carboxylase enzyme, as you can see. The product will be an oxaloacetic acid that has got 4 carbons. Hence, a C4 pathway. The first compound to be formed in this pathway that is stable has got 3 carbons, whereas in a C3 pathway, the first compound that is formed that is stable, which is a PGA, has got 3 carbons. Here in this case, OAA has got 4 carbons, hence a C4 pathway. The next step is this OAA, which is being formed within the cytosol of the mesophyll cell, will be delivered into the chloroplast of the mesophyll cells. There you can see going into the chloroplast, this green thing inside the mesophyll cell is your chloroplast. Inside the chloroplast, this oxaloacetic acid is going to be converted into another organic acid which has got 4 carbons, and this organic acid is called malate. The conversion of OAA to malate is done by an enzyme called Malate Dehydrogenase that you can see there. Still, the malate that is formed, it's a 4-carbon organic acid. This is what happens within the mesophyll cells. The next step then is going to be, you can see this malate is delivered from the chloroplast of the mesophyll cells through, we can say, the plasmodesmata, is delivered from the mesophyll cells into the chloroplast of the vascular bundle sheath cells there. Here. Inside here, this malate will be decarboxylated, which means it is going to be broken down into carbon dioxide and a pyruvic acid or pyruvate there. So you are breaking this malate into a CO2 and a pyruvate. So this compound had 3 carbons. There is a CO2 having 1 carbon. Therefore, it means the pyruvate will be having 3 carbons. That pyruvate is delivered back into the chloroplast of the mesophyll cells, as you can see, and it is converted into PEP by the enzyme called pyruvate phosphate dikinase. You can see the name of that enzyme there. And then the next thing then is this PEP is delivered out of the chloroplast into the mesophyll cells, and it is ready to combine with another incoming bicarbonate or another incoming carbon dioxide. So we are done with what happens in the mesophyll cells. Let's come here. Now, there is the carbon dioxide that has been released from the breaking down of the malate. That carbon dioxide, as you can see, enters the Calvin cycle, which means this carbon dioxide now combines with RBP catalyzed by the enzyme called RBP carboxylase, abbreviated as Rubisco. And the Calvin cycle, as you know it, as we have dealt with it before, takes place, and that is going to result in the formation of glucose molecules, the regeneration of the RBP. The Calvin cycle, the way you know it, takes place within the bundle sheath cells. So you can see in this pathway, carbon dioxide is temporarily fixed within the mesophyll cells. At this point, there is no sugar that is formed. In the mesophyll cells, only organic acids are formed. These organic acids are just compounds that are used to hide the carbon dioxide. And these organic acids are delivered through some kind of a pump, an active process, into the bundle sheath cells. And inside there, they are broken down to release the carbon dioxide and these 3 carbons containing compounds. I forgot to mention to you that the enzyme that decarboxylates the malate is called NADP malic enzyme here. That's very critical. You must know these enzymes. Now, the Calvin cycle takes place with the use of the CO2 which is coming from the atmosphere, by the way. You can see it's blue, still blue, the same CO2, we're tracing it. The CO2 is hidden on the OAA, still hidden in malate. Malate is delivered there. The malate is broken down. The CO2 is released. You can see it's blue. It's the same CO2 that came from the atmosphere. Eventually, that CO2 is delivered towards the Calvin cycle. It's going to form the glucose molecules, which can be linked to form starch and so on and so on. Some of them can be converted to form sucrose and so forth and so forth. So basically, that is your typical C4 pathway taking place in C4 plants. An example of such kind of plants, it's a sugar cane. Sugar cane is a C4 plant. Another nice example of a C4 plant, it's maize plants. The milli-mill plants that you all know, it's a maize plant. So a typical C3 plant, it's plants like wheat. Wheat is a C3 plant, whereas maize is a C4 plant. Sugar cane is another example of a C4 plant that fixes carbon dioxide through a C4 pathway. So in the next clip, I'm going to try and explain the advantages of this pathway. In the next clip, I'm going to focus on the advantages of this pathway. Thank you.